Audio Jungle. Audio Jungle.
In the respiring tissues, A. Carbon dioxide release diffuse into the red blood cell. B. In the red blood cell, carbon dioxide fuses with water to form carbonic acid with the help of the enzyme carbonic anhydrase. C. Carbonic acid dissociate into H+ ions and hydrogen carbonate ions. Hydrogen carbonate ions diffuses out into the blood plasma. D. Chloride shift occurs to maintain electrical neutrality. Chloride ion diffuses into the red blood cell. E. Oxyhemoglobin dissociates to release oxygen to the respiring cells. H+ ions will then combine with hemoglobin to form hemoglobinic acids. In the lungs, A. Hemoglobinic acid dissociate to form H plus and hemoglobin. Hemoglobin acquire oxygen to form back oxyhemoglobin. B. Hydrogen carbonate ions in blood plasma diffuses into the red blood cell. C. H plus ions and hydrogen carbonate ions combine to form carbonic acid. D. Carbonic acid dissociates to form carbon dioxide and water with the help of the enzyme carbonic anhydrase. E. Carbon dioxide is then diffused out through the lungs through exchange of air. The Bohr effects. The Bohr effects describe how carbon dioxide and H plus concentrations affect hemoglobin's affinity for oxygen. An increase in carbon dioxide in blood results in the reduction of oxygen carried by hemoglobin. The Haldane effect. The Haldane effect describes how oxygen concentration determines hemoglobin's affinity for carbon dioxide. An increase in oxygen carried by hemoglobin results in less carbon dioxide carried by carbaminohemoglobin. When the first oxygen molecule is bound to a hemoglobin molecule, it changes the conformation of the hemoglobin molecule and facilitates the second and the third oxygen to bind. The binding of the fourth oxygen is not facilitated, so there is a slight delay. This is also called molecule cooperation. When one mole of oxygen binds to one of the heme group in the hemoglobin, produces a conformation change in that subunit, transmitted to the other three subunits, facilitate oxygen binding to the rest of the polypeptide. Concerted conformational change of the subunits increases affinity of hemoglobin for oxygen, resulting in sigmoid shape for the oxygen dissociation curve of the hemoglobin. The cooperative effect is reversible. When one subunit of the oxyhemoglobin unloads its oxygen, other three quickly follow suit. Conformational change lowers its affinity for oxygen. Transport of oxygen. Number 1. 98.5 of oxygen transported by red blood cell. Red blood cell contains the pigment hemoglobin which carries the oxygen. Number 2. In the lungs, oxygen diffuses from the alveoli into the blood in the capillaries because of lower partial pressure of oxygen. Number 3. Oxygen diffuses into the red blood cell where it binds to each of the Fe2 plus ions in the 4 heme groups in the hemoglobin to form oxyhemoglobin HbO2 4. So, each hemoglobin can carry 4 oxygen molecules. Number 4. Oxygenated blood transported to the heart from the lungs by pulmonary veins and pumped out of the heart through aorta and arteries to supply all parts of the body with oxygenated blood. Number 5. When it reaches a tissue which needs oxygen, the oxyhemoglobin releases its oxygen to tissues because of lower partial pressure of oxygen. This is due to H plus ion released when CO2 is converted into carbonic acid. H plus react with oxyhemoglobin, releasing the oxygen. Number 6. The oxyhemoglobin then becomes hemoglobinic acid. The blood is said to be deoxygenated. Comparing maternal and fetal hemoglobin oxygen dissociation curve. A. The curve shift to the left, still sigmoid curve. B. Fetal hemoglobin has a higher affinity for oxygen. C. So that at low partial pressure of oxygen, fetal hemoglobin is saturated with oxygen. D. Oxygen is readily dissociated from the maternal to fetal through the placenta. E. The fetal hemoglobin has a slight difference in structure due to no direct contact with oxygen and rapid metabolism and growth in fetus. Comparing myoglobin and hemoglobin oxygen dissociation curve. The curve shifts to the left. The curve is hyperbolic. B. Myoglobin has a higher affinity for oxygen. C. So that at low partial pressure of oxygen, myoglobin is saturated with oxygen. D. Oxygen is readily dissociated from the hemoglobin to myoglobin. E. During strenuous exercise, myoglobin able to replenish the oxygen stored in muscle cells to prevent anaerobic respiration. Comparing llama and humans hemoglobin oxygen dissociation curve. A. The curve shift to the left, still sigmoid curve. B. Llama hemoglobin has a higher affinity for oxygen. C. So that at low partial pressure of oxygen, llama hemoglobin is saturated with oxygen. D. Higher affinity facilitates the oxygen uptake. Even at low partial pressure of oxygen, at higher altitude. Extra. Comparing mouse and human hemoglobin oxygen dissociation curve. A. The curve shift to the right, still a sigmoid curve. B. Mouse hemoglobin has a lower affinity for oxygen. C. So that at low partial pressure of oxygen, mouse hemoglobin is not saturated with oxygen. D. Mouse being small in size, large surface area per unit volume ratio has a higher metabolism to generate heat. So, to quickly unload, to keep the tissue supplied with oxygen. Oops. Regulation of human breathing. Partial pressure of carbon dioxide in the blood increases. It is detected by the chemoreceptor. There are two chemoreceptors, a peripheral chemoreceptor, which are aortic and carotid bodies, detect changes in partial pressure of carbon dioxide, H plus concentration, and partial pressure of oxygen. B. Central chemoreceptor or medulla detects changes in partial pressure of carbon dioxide and H plus concentration. Impulse is relayed to the breathing control center in the medulla oblongata. Inspiratory center sends out impulse to the diaphragm muscle through pharyngeal nerve and external intercostal muscle through external intercostal nerve. Lungs expand as inspiration takes place. Then, stretch receptor at the wall of alveoli and bronchioles are stimulated. Impulse is relayed to the expiratory center through the vagus nerve. Automatically inhibits the inspiration center and inspiration. Lungs deflate as expiration takes place. Expiration is a passive process. When stretch receptor no longer stimulated, expiration center becomes inactive and inspiration begins again. Normal breathing rate 12 to 15 breaths a minute. the structures of stomata. A. Thin outer cell wall of gut cells and thick inner cell walls of gut cells. B. Both gut cells are connected at the ends, forming a pore, stoma. C. Cellulose microfibrils is radially arranged. D. Kidney bean shape, has chloroplast and surrounded by subsidiary cells. Potassium ion accumulation hypothesis. Opening of stomata. A. In the presence of blue light, photosynthesis takes place. B. ATP phase, proton pump is activated, actively transporting H+ ions out from the gut cells. C. Potassium ion diffuses into the gut cell by facilitated diffusion. D. Solute potential increases in the gut cell. Water potential decreases in the gut cell. E. Water diffuses into the gut cells by osmosis. Gut cells become turgid. Stoma opens. Closing of stomata at night. A. At night, ATP phase, proton pump is not activated. B. Potassium ion diffuses out of the gut cells by facilitated diffusion. C. Solute potential decreases in the gut cell. Water potential increases in the gut cell. D. Water diffuses out of the gut cells by osmosis. Gut cells become flaccid. Stoma closes. Closing of stomata during drought. A. Abscisic acid ABA synthesized by mesophyll cells binds to the ABA receptor. B. The binding causes the influx of calcium ions into the gut cells. C. Proton pump is inhibited. D. Potassium ion diffuse out of the gut cell by facilitated diffusion. E. Solute potential decreases in the gut cells. Water potential increases in the gut cells. F. Water diffuses out of the gut cell by osmosis. Gut cells become flaccid. Stoma closes.